Alright, so welcome back. So in our last discussion, we're talking about the diakinesis, right? The nuclear membrane breaking down, nucleus disappearing, the central getting to the opposite pole of the cell, alright? So now we talked about the metaphase 1, where the central form the spindle fibers, alright? So when it forms the spindle fiber, what happens? The homologous pair of chromosomes, each of them are already bivalent, right? You know that. So the homologous pair now, they come to line along the equatorial plane of the spindle fibers. So you can see them lying along the equatorial plane of the spindle fiber. So that's what happens at metaphase 1. At anaphase 1, what happens is this. Remember at during mitosis, normally it usually divides at the centromere, right? The chromosomes, there's usually a division at the centromere, but here now at anaphase 1 there is no division at the centromere. So what happens is this: the sister the chromosomes now, the chromosomes themselves that form the homologous pair, they just separate. No division at the center, they just separate and then they migrate to the opposite pole. Alright, so they just separate from each other and migrate to the opposite. You can see it before they exist as one homologous pair, but they just separate here and migrate to the opposite poles. So at the end of the day, you now have this cell. You have this cell that has 23 bivalent chromatids. Alright, so that you can see 23 paired chromosomes. Then you have this cell having 23 bivalent chromatids or 23 paired chromosomes. Alright, because the chromatids will later become chromosomes. They are chromosomes. Alright, so now telophase 1. So what happens at telophase 1? The chromatids now, the bivalent chromatids now have gotten to the end of the opposite pole. Alright, they've gotten to the ends of the opposite pole. So this is what happens now. The spindle fibers will break down. So this, you can see the spindle fibers, they're already breaking down. The spindle fibers will break down. Then you begin to observe that the nuclear membrane begins to reappear. It reappears, and then the nucleus will reappear at this telophase 1. So once that occurs, you can observe that a constriction begins to set in. There's this constriction that's setting. Alright, so that's what happens at this telophase. The spindle fiber breaks down, the nuclear membrane reappears, nucleus reappears, and then the constriction occurs. Alright, so now let's look at what happens at the next phase, which is cytokinesis 1. Remember we said that division is divided into karyokinesis and cytokinesis. Karyokinesis is division of the nucleus of that cell, while cytokinesis is division of the cytoplasm. So after the cell has gone through karyokinesis, has entered at this telophase 1, it's going to go into cytokinesis 1, where the cytoplasm divides. So there will be remember you have this constriction here, so a cleavage will occur at this constriction. So the result of this cleavage at this point of completion is two daughter cells that are non-identical. Why do we say it's non-identical? The reason why we say it's non-identical is because remember at prophase, at diplotin, alright, there's something that occurred there, crossing over, exchange of genetic material. So because of that exchange of genetic material, that crossing over that occurred there, the cell itself will not be identical because the chromosomes, they will not have similar characteristics. Like this A, the chromosomes here in this cell A will not have the same characteristics as the chromosome here in this cell B. So the cell will differ in characteristics, in their functional characteristics. Alright, they will differ. So that's why it will be two non-identical daughter cells formed, and these daughter cells will have 23 paired chromosomes in each of them. That's 23 bivalent chromatids in it. Alright, so now let's go into the second meiotic division. So in second meiotic division, it also has interphase. Interphase is just a phase existing between the end of one and the middle of another. So it's starting from prophase. Alright, so but it's going to begin, this cell will go into interphase 1 first. So but here at this interphase 1, no duplication of DNA is occurring. Alright, why? Because the reason for duplication is to form bivalent chromatids, right? But the cell already has bivalent chromatids, so there is no need for DNA duplication to occur, alright, because we already have a bivalent chromatids. So no duplication occurs here. So the cell now goes into prophase 2. So this is just to show you the difference between prophase 1 and prophase 2. You can see here you have the tetrads, right? The homologous pair, which are making up bivalent chromatids for one each of them. So here at prophase 2, what happens is this: the chromosome becomes rod-like, as it is usually is rod-like on the norms, alright. But at late prophase 2, what happens is this: the nuclear membrane breaks down, alright, and the nucleus disappears. That's what will occur at late prophase 2. Then the centroids now, they begin to migrate to the opposite pole, and as they migrate to the opposite pole, they form microtubules, and these microtubules are what will form the spindle fiber. So the spindle fiber will then run from the one central here to the other at the other end. Alright, so that's what occurs at prophase 2. So now what occurs at anaphase 2 is this: the central has formed spindle fibers already, the nucleus and nuclear membrane has disappeared, so the nuclear content is floating in the cytoplasm, alright. So what happens is this: the bivalent chromatids, you can see it now, this one cell and this another, the bivalent chromatids, they come to line along the equatorial plane of the spindle fiber. Just remember one thing, that this second meiotic division is similar, is very similar to mitosis, alright. So the second meiotic division is similar to mitosis. So if you notice, that's what's happening. So you will see the bivalent chromatids coming to line along the equatorial plane at metaphase 2. So at anaphase 2 now, what happens is, what happens at mitosis will occur here. Look at it. There will be a splitting longitudinally at this centromere. So the centromere will split longitudinally, alright, so that these bivalent chromatids, they will separate. So you will have a single chromatid that will become a chromosome, and a single chromatid that will become a chromosome, alright. So as they divide here and they separate, they start moving to the opposite pole at anaphase along the spindle fibers. So that's what occurs at anaphase 2. So a splitting will occur, and then it migrates to the opposite pole. So at telophase 2, they finally get to the opposite pole, and when they get to the opposite pole of the spindle fiber, the spindle fibers will break down, and then the nuclear membrane here and the nucleus will reappear, alright, and then all cell organelles, remember at telophase, even at telophase 1, all cell organelles will duplicate, and the reason for that is to ensure that each cell gets the same number of the cell organelles that exist in the original cell, that's the parent cell. So all cell organelles will duplicate, alright, so that this cell that is to be formed will have the same cell organelles as this. So the only thing that will differentiate them is their genetic makeup, alright. It's genetic makeup, and genetic makeup will now differentiate them by the proteins that we have, alright. So because genetic makeup, DNA, is what will enhance protein synthesis, and that will determine the function of that cell. So that's the general thing that this genetic makeup, the DNA, does. So if there's a difference, there will be difference generally in the cell, alright. So at telophase, this is what will happen. So now we have cytokinesis 2. At cytokinesis 2, here you have this at telophase, there is this constriction, alright. So there will be a cleavage at this constriction at cytokinesis 2. So at cytokinesis 2, when that cleavage occurs at the point of constriction, you form two non-identical daughter cells. So in total now, you are having two non-identical daughter cells formed from the first meiotic division, and two non-identical daughter cells formed from the second. So from the first, you're having two, and those two, they're having 23 paired chromosomes, that's 23 bivalent chromatids. Then each of those cells will go into division, so that at the end, when there's a splitting at at anaphase, all right, the splitter and the node, you will now have you now have cells, two cells from each, two cells, that's 4 cells altogether, having 23 single chromosome, 23 single chromosome, 23 single. The 4 we have 23 single chromosomes. So in total you have 23 number of chromosomes. That's the haploid number of chromosomes as compared to the parent cell, which has diploid number of chromosomes. Alright. So the reason for that is at fertilization, the sperm cell, the matured oocyte, they come together and there will be a fusion of this nucleus. And when there is a fusion, the 44 number will be restored, the diploid number will be restored. Alright. So that's it for this class. So it's good to have you guys around. Ensure that you are following the classes in sequence. Alright, so when you just type in Anatomy Book Club, all you need to do is to go to the playlist. So in the playlist, you can have several videos here. You have the back, upper limb, lower limb, head and neck, abdomen, thoracic embryology, neuroanatomy, pelvis and pelvis. So what you need to do is to just click on the embryology. You don't need to watch the whole video. So you just click on the embryology. You just click on the embryology. You just click on the embryology, and then you'll be able to access all the videos there. Alright, you'll be able to access all the videos in the embryology playlist. So there are videos there in embryology playlist. So you just access them, and they are arranged in sequence. If you notice, they are arranged all in sequence. Alright, you have introduction first, you have your cell division, then go into garment formation and the rest. Then one thing is this: we'll continue to upload content consistently from henceforth. So ensure that you subscribe to the channel, and then make sure you turn on your notification. That's the most important thing, so that once videos are uploaded, you get notified. Alright, so for those who have not joined our other platforms like our Telegram, where we post MCQs, past questions, we brainstorm, and then our Facebook page where we post relevant content, and our WhatsApp TV where we post forms, memes, and anatomy concept. So ensure you visit all these platforms and get the most out of Anatomy Book Club. So it's good to have you guys around. So this is Sir Lawrence. So in the next class, we'll be talking about cell cycle and chromosomal abnormalities. So good to have you guys. Have a great day.